presentation here. I just wanted to thank Jack for inviting me to come and visit and present here today, and to the organizers who put this conference together. It's an interesting collection of concepts being explored here, and it's a fun meeting. And certainly I wanted to thank Kristen for all of the work she did in terms of helping me get here in a reasonable order. So I just wanted to make my disclosure here. I'm a founder of two companies, both of which are involved in using different herpes simplex viruses to treat patients with different types of tumors, but mostly with brain tumors. So the talk is actually about using mouse avatars, but I'm going to talk towards the end of provide information towards the end of the talk about our experiences with using human herpes viruses and how the information that we're deriving from the mice can help us decide which patients would be treated and which ones would not. So I clearly am just sort of the general supervisor. I stand back and let most of these folks do the heavy lifting, and I'm pleased that they do such a great job at heavy lifting. The problem that we're facing with primary brain cancer is the incidence is relatively small. So Anna Barker at the NCI classified this as one of the little tumors as opposed to one of the big tumors. And essentially the issue is that most people don't think too much about brain tumors from the standpoint of the numbers of people who are affected, but I would venture to guess that most of the people in this room know someone, either a celebrity or a family member or a relative or a colleague or friend who has had a diagnosis of brain tumor. The problem with most of these tumors is that there's a very high mortality incidence. 25% of the childhood cancers are brain tumors, and it is the leading cause of cancer death in children. But again, the numbers are small. Approximately 3,600 children in the U.S. will develop a malignant brain tumor this year. And most of those are not curable, and these children will die. 60% of primary intracranial malignant tumors, and I'm not including meningiomas and other types of tumors that occur within the calvarium but are not intrinsic brain tumors. These are high-grade malignant gliomas. They're derived from a glial progenitor cell of some sort. It could be a progenitor cell that would eventually develop into an astrocyte, or one that would eventually develop into an oligodendroglial cell. Brain tumors are the third leading cause of death. So this is an important medical problem and clearly an unmet need. These statistics really haven't changed very much in the last 75 years. So we've gained about 1 month of median survival in the high-grade glioma field for every decade of research. So we're not doing a very good job. And part of the problem that we're not doing a very good job is that we don't know that much about them, and we're learning more now. In the last 10 years, there's been an explosion of information about brain tumors. But here again is the distribution that one would expect to see, in the sense that low-grade tumors are really not a very large proportion of these. At diagnosis, the grade 3 and the grade 4 tumors are the ones that actually present as a clinical malignancy. For a very long period of time, I would say since 1978, the major predictors of survival for patients with brain tumors has been pathology. The worse the pathology, the worse the patient does. Age, not surprisingly, the younger the patient, the better they do. Neurological function status and the extent of resection. The problem is that these tumors, unlike most other types of tumors, don't metastasize, and it may be that they really do metastasize, but the patients don't live long enough for us to detect the metastasis. But in a few cases, we've found metastatic lesions in the patient population. Survival curves are very depressing, and again, this slide is from 1978, and the statistics really haven't changed very much. It's about the same. If you look today and do the same type of Kaplan-Meier survival curve. So the problem is that the tumor occurs in a site where it's difficult to remove all of it. You can remove some of it, but maybe take the music lessons, but you want to leave the ability to walk and talk. So the essential problem is that there's a lot of tumor left behind, and generally the tumor that recurs within 2 centimeters of the margin of resection. So if you think about this, the tumor cells have invaded out into normal brain. They are very invasive cells, and so the tumor recurs locally, but it can also recur distantly. That makes brain tumors or malignant gliomas a whole organ disease. So we have to think about brain tumors as being a whole organ disease, not just a spot or a lump that we can see within MRIs like this, or when the patient's head is actually opened up and the tumor is visualized. More importantly, malignant glioma cells are resistant to conventional therapy, and this may be part and parcel of their call in life. Astrocytes are designed to protect and serve the neural elements within the brain. And so the concept is that they scavenge things that are bad, they produce things that are good for neuron survival and development, but they essentially inherit and turn to a very poor purpose those same properties. As these tumor cells invade into normal brain, they are protected by the blood-brain barrier. So delivery of drugs to the tumor is impaired by this fact. We can get drugs into this component here because, as you can see in the MRI, it's very, very leaky. It's a very highly vascular tumor, and the contrast medium has leaked out of the surrounding blood vessels and leaked into the tumor mass. So we can see the tumor mass, but what you can't see is around the tumor mass there are cells that have not yet influenced the vasculature to be leaky, and as a consequence, those cells are protected. The other problem is that less than 12% of the cells within these tumors are proliferating at any one time. So if you give them or treat the patient with an agent that is essentially a DNA alkylating agent or some other agent that attacks cell proliferation or the mechanism of cell proliferation, more than half, usually 70 or 80% of the cells are totally unaffected. Radiation is effective. It's the single most effective therapy that we have. So the problems that we are facing is that the traditional therapeutic tripod for treating brain tumors are really surgery, radiation therapy, and it's usually a pretty high dose. It has worked its way up from 54 gray to about 60 gray, delivered in 30 fractions over a 6-week course, 5 days a week. Patients are not treated on the weekends because it's not convenient. They receive 1.2 to 2 gray a shot, so it's not a very big dose for each individual fraction. But over the cumulative effects of the radiation are really pretty good. The best drug that we have right now is temozolomide, and this is an alkylating agent. It replaced back in the, I would say back in the 2000s, it replaced BCNU, which for, I would say 30 or 40 years was the drug of choice. BCNU was a very nasty drug. It had to be given intravenously. It was highly emetic, and basically patients hated it. Temozolomide is an oral drug. They take a few pills a day. It's a much better therapeutic from that standpoint. Generally, chemotherapy, the temozolomide is given concurrently with radiation, and that has been found to increase the median survival by 2.5 months. So that is now the new standard of care. We've gone from approximately 12 months median survival to about 15 months median survival. Temozolomide is given as a maintenance therapy after chemoradiation. but that may or may not have any benefit. That really is sort of the standard that folks use. It's basically we don't have anything better, so we're continuing to use the same thing that we used or have been using. So as we develop emerging technologies in terms of new therapies that would be used, we have to answer the question as whether or not there will be improved benefits with acceptable risks for these new therapeutics that we're going to be using. And again, the challenges that we face are really delivery, specificity, and efficacy of the agent that's being applied. So as we build on this particular tripod, we have a lot of new ways of doing things. Surgeons are now much more involved in the therapeutic aspects of treating patients with brain tumors. They're putting in catheters and delivering drugs and other agents, such as viruses, by a mechanism called convection-enhanced delivery. We're using radiation more as a means of enhancing drug therapy, different types of drugs, and also for enhancing virus therapy. One of the things that most viruses like to do is they like to modify the cell so that it suits their purpose in terms of the virus being able to replicate. And one of the ways they do that is to induce a DNA repair response. So radiation will mimic that DNA repair response and make the viruses that are being used much more effectively because it sets up the proper soil, as it were, for the virus to replicate. And then chemotherapy is really a burgeoning area right now. There are a lot of small molecule inhibitors that are being developed by the various pharmaceutical companies. They're given orally for the most part, but the big problem there is only about 1/3 of them actually partition into the CNS. So again, the blood-brain barrier blocks the entry of these particular molecules. Nanoparticles are being developed, apoptosis inducers, and of course vectors for immunotherapy, and this may apply to the fourth talk of the afternoon, and so I'll mention a little bit about that. The elephant in the room that we really are not addressing here is whether or not we can use some sort of discernible feature of a specific high-grade glioma specimen and translate that into information that the clinician can actually use to plan for the patient who's going to be in the clinic in the afternoon. And so the problem that we have is one of being able to get the information about the tumor to the clinician and in a form that makes sense, and that decisions can be made as to how the therapy should be applied. One of the biggest things that has really driven our understanding of brain tumors has been the Cancer Genome Atlas project. Now, this is a $200 to $250 million a year program that was set up by the NCI several years ago. They started out with a 3-tumor pilot study, which was about $225 million to do this. And essentially, they looked at these 3 tumors. GBM was first. Squamous cell carcinoma of the lung was the second tumor that they looked at. And serous cystic adenocarcinoma of the ovary was the third. And TCGA essentially used the molecular techniques that were available at that time to make all of these types of determinations. And so you talk about big data, this generated big data. And they didn't do it through a single system, but they actually did it through a number of different contractees. So the Biospecimen Core resource went out and recruited more than 150 tissue source sites. There were 6 different cancer genomic characterization centers, places like the Broad. There were 3 genome sequencing centers, places like TGen. There were 7 genome data analysis centers, some both in TGen and places like the Broad and other places. And then there was a single data coordinating center. And generally what they developed was an enormous amount of information about GBM first, and then lung cancer, and then ovarian cancer. They're now moving towards over 25 different forms of cancer because of the findings that were made in this particular pilot study. So the question that arose is that even after they profiled very stringently over 500 GBM tumors, and the goal is 500 tumors of every type, how do you use the data? So when they initially analyzed the platform, we learned a lot of things we didn't know, some of which we suspected, but we were not clear on. There were at least 4 different molecular subtypes of GBM. If you look at them histopathologically, they all look the same. There's no way to differentiate one from the other. There were very clearly defined dysregulation of pathways that drive cell proliferation and that drive other properties that contribute to tumorigenesis. And finally, there were very specific pathogenic genetic mutations. There were pathogenic gene expression profiles that were different, and many other biomarkers suddenly became evident through the analysis of these data. And again, the data looked at DNA, it looked at RNA, it looked at microRNAs, it looked at a number of other properties of these cells in tumors. So in order to use this data, we need to develop some sort of appropriate experimental system so that we can test our hypotheses about the disease itself and also any emerging treatments that might be appropriate. And the requirements for this type of experimental system is that we really have to have a fairly realistic biological model for GBM tumorigenesis. We have to have a model that really is more patient-like than the things that we've been doing in tissue culture, where we're growing cells in a dish in the presence of 100% or 20% oxygen and with growth factors and a lot of other things lots of high glucose that allows these cells to grow at the fastest speed possible. This also reflects the diversity of, it should also reflect the diversity of tumor subtypes. So in GBM, we would expect to find the same 4 molecular subtypes that have been previously defined. And it should be a model that allows for timely testing of either the hypotheses or the drugs. And the model that we came up with in 2005 when the TCGA first started was to actually look at GBM tumors that were implanted directly from the brain of patients into mice. And the idea being that if we passed them into tissue culture, we would have the same problem with tissue culture that exists today with all of the established cell lines that are out there that have been cultured for 40 years or more. If we put them into the mice, the tumors would have to struggle to develop and survive. They would face a lot of the same problems with access to oxygen, access to glucose, and just being in a slightly different environment or a greatly different environment. But we could pass these, establish these tumors. It takes 2 to 3 months to do this, so they don't grow right out of the mouse. They take a while to adapt and to realize where they are and to develop into a tumor that you can then harvest and pass to the next mouse in the series. You can use those cells for intracranial studies and putting the tumor in a orthotopic location rather than in a heterotopic location. And we can even harvest brain tumor initiating cells, or sometimes called glioma stem cells, directly from these tumors because they were in the original patient population. They remain in there. In fact, everything in the tumor that you put into the mouse remains in the tumor that you pass to the next mouse. So it's much more patient-like than taking disaggregated cells and putting them in tissue culture with lots of growth factors, lots of oxygen, lots of glucose. So these lines these xenolines that we develop actually retain a lot of the realistic environment for tumor growth. They essentially are subpopulations. It allows for subpopulations within these tumors to develop, but they're not under strange selection problems. They're under selection problems in vivo that they've experienced in patients. An example of that is the expression of epidermal growth factor receptor variant 3. This is a mutant version of epidermal growth factor receptor, but it is constitutively turned on. However, if you put those cells into tissue culture with medium that contains fetal bovine serum, the platelet-derived growth factor will actually cause the cells that do not express this particular receptor to actually overcome the population, and within a few generations you've lost all of the EGFR variant 3 cells, and so they disappear from culture, but they are retained in these model systems. The other important thing is things like invasion are retained. This is a glioma cell line that's been in culture since 1968. It was put into culture from a patient's tumor by Jan Pantin at the University of Uppsala. And if you inject those cells into a mouse, it grows like a rock, a lump. GBM X gliomas, or the PDXs we call them, the patient-derived xenolines, essentially retain the invasive characteristics that you see here, the same types of invasive characteristics that are seen in patients. So this line is much more important in terms of their behavioral characteristics, especially if you're looking at invasion or other types of properties that require migration. These guys don't invade at all. So it creates a more realistic environment. You can do short-term cultures. You can, once you've established it, you can do multiple experiments relatively rapidly. You can expand these lines very easily in animals. And then you can do testing of drugs using what we call a proband model. Now, it's impossible to harvest a tumor from a patient and put it into these mice and then 3 or 4 months later start testing that tumor. It's really difficult to do that. By then, the patient is almost beyond help, and the issue is, well, how do we overcome this? And one of the reasons, or one of the ways that we've thought about overcoming this is the fact that we will use a proband model. In other words, we will produce a panel of tumors and use those as a panel against which to compare individual patients' tumors as they come into the clinic, and then essentially determine which treatment is best. And so it is sort of described, the proband model is sort of described here, where the first stage is perhaps the more difficult stage. You first have to establish the panel and validate the panel. You molecular profile the tumors to make sure that they match up with what has been determined. You can actually then do drug regimen profiling with whatever drugs you think might work, determine the response, again repeat the profiling to see how the tumor has changed, and then establish an order of eligibility. At the same time, you would then provide some profiling of patient tumors as they come into the clinic. This molecular profiling then would be compared with what we have done with the tumors. We would try to match a patient's tumor with one or more tumors growing in the mice from a different patient, obviously, and then make decisions as to how the eligibility criteria defined for this patient-derived xenoline would then apply and make decisions as to which drugs would be most appropriate for the patient whose tumor is being profiled here. And so the idea is that this proband model provides a mechanism, but as you can see, it takes a while to get from this point to this point because it's fairly convoluted. Nevertheless, we think that it has a pretty good reason for working, and that's essentially what I wanted to talk about today. So we started our panel back in 2005. We currently have about 42 of these xenolines from 42 different patients. It takes 2 to 3 months for these to develop. We can keep them in low passage. We freeze them back so that we can actually go back to a lower passage every month or so and start fresh with that particular tumor. We've characterized 27 of these. We can only afford to characterize 27 of these. It cost us about $890,000 to characterize these 27 tumors. We used 4 different arrays. We used an Omni 2.5 million SNP bead array produced by Illumina. This looks at copy number variation and other genomic changes that occur. We selected 40 genes that are known to be amplified, deleted, mutated, or changed in some way in GBMs, and we did deep sequencing to see what kinds of changes we could detect that way. We did Affymetrix whole transcript human exon profiling of the gene expression. So DNA is converted into RNA. RNA is the instructions for how a protein is assembled in the cytoplasm of the cell. And essentially, this type of array tells you what RNAs have been transcribed from the DNA gene. And a lot of times, most people think, I suppose, that all of the genes are being transcribed at some point or another, and that's not the case. Very few genes are actually being transcribed. But this will actually look at every gene, every RNA that has been produced. If the RNA has been produced, there is a good chance that the protein might be produced. But if the RNA is not there, even though you might have a genomic change within the tumor, it might not have any bearing on what that cell or what that tumor is actually doing. Then we did something that's different, something that the TCA did not do, and we did whole kinomic profiling, and I'll describe that in just a few minutes, but it basically is looking at enzymes that phosphorylate proteins on tyrosine amino acids or on serine threonine amino acids, puts a phosphorus on there. That doesn't seem like much, but it means a whole heck of a lot to the protein. It either silences the ability of the protein to work, or it activates the protein. So it can be one of 2 different things. And really, all of the action that's going on inside of a cell is going on inside of the cell because of proteins. It's not going on inside of the cell because of what the genes are doing. It's just simply the genes are being transcribed to RNA, which is then maybe or maybe not producing the protein, which is then the actor, which actually produces the phenotype that we see in tumor cells. So the questions we wanted to ask, if we compare our xenolines with the TCGA, do we see the same molecular subtypes? Are there changes? What are the differences? And is one method of carrying the xenoline better than another? Is the orthotopic method of putting the tumor cells in the brain better than putting the tumor cells in the flank? Now remember, these are brain tumors, all right? So just to show you a heat map of the data that we've generated, if you look over on the left-hand panel here, these are the signature genes. There are 840 of them, 210 for each of the 4 molecular subtypes. And without really looking too hard, you can see that there are 4 different patterns of the gene expression. This is the proneural, this is the neural, this is the classical, and this is the mesenchymal subtype. So 4 separate subtypes in the TCGA data set. If you look at our data set over here, we see the same types of separation. Some of the genes are up or down slightly differently, but for the most part, we have proneural, we have neural. The largest group is the classical, and then we have mesenchymal tumors over here. So we see essentially the same types of gene expression profiles that were seen in the patient's tumors by TCGA. So we can classify our 27 tumors as such, in which we look at the classical tumors, of which we have the most. The neural tumors, we only have 3 of those, the proneural tumors and the mesenchymal tumors. And we know how quickly these tumors grow in the mice, and these were all done by putting tumor cells in the brain of the mice, allow the tumor to grow up to a certain size, kill the mouse, took out the tumor, and then processed it or analyzed it. So we did not put them in the flank and grow them in the flank. We actually grew these orthotopically in the brains of mice. So you have to do a lot of mice in order to get enough tissue to do the analyses. One of the things that popped up when we started looking at our data in a little bit more detail was that if you compare the gene expression profile in the TCGA, so we're looking just at the gene expression profile, and these are tumors from patients, as well as they did about 23 subcutaneous PDX, but that's not included in this analysis. If it's included here. If you look at the gene expression profile compared to a gene expression profile developed for GBM cell lines separately from the TCGA, what you see is that you would expect, if you're looking at fold changes that occur in these genes, you would expect that each one of these dots for both the cell lines and for the primary tumors to fall along this diagonal line. Well, about 70% of the genes do not. And basically those are genes that are different from the cell lines and the patient's tumors. So I don't know what we've been studying for the last 70 years of doing tissue culture with tumor cells, but it looks like this is quite a bit different in terms of the gene expression profile than what one would expect to see in the patient tumors themselves. If you take the tumor cells or tumors, the subcutaneous tumors, and compare those with TCGA, it gets a little bit better. The genes that stand out as being different down here, all of these are host immune response genes. These are genes that are expressed by lymphocytes and macrophages from the human patient. You would not expect to find lymphocytes and macrophages from human lymphocytes and macrophages in the mouse. So those appear down here. The genes that appear up here as being increased in the subcutaneous tumors are essentially those that are dealing with cell proliferation and DNA repair. And so these are actually a little bit more amplified in the subcutaneous tumors than they are in the patient's tumors. However, if you look at the tumors that are grown in the brain and compare that gene expression profile and this gene expression profile, the GBM or the TCGA data are here, and the orthotopic PDX data that we developed are here, you can see that the only thing that really stands out as being different are the human lymphocyte and macrophage genes, and you wouldn't expect to find those in the mice. So it looks like the orthotopic system is better, but in a pinch, the subcutaneous is clearly a lot better than doing the cell line types of studies. We've also looked at copy number variation, and we see that there are specific things that are upregulated. For example, in chromosome 7, we know that in many of these tumors, especially the classical tumors, there's an amplification of the gene for EGFR, the epidermal growth factor, and it's right here. In chromosome 17, there are mutations and deletions that occur in P53, and that's located right here. So it drops below the line, this goes above the line. And if we compare for one cell line across all of the linkage groups from chromosome 1 down here to 22, we see that the profile shows that amplification in 7, and it shows diminution in 10 and a partial diminution in 17. And if we look at all of the tumors and compare those to what was found in the TCGA, we see that the major amplification we see in chromosome 7 is replicated here in the TCGA profile, and the deletions that are seen in, say, chromosome 10 in patients, which contains the gene for P10, are also seen in some of the tumors that we have looked at in the PDX samples. So genomically, our tumors look very similar to the characteristic signature that is seen in human GBM tumors. And the copy number variations that have been found in these primary GBM samples in the TCGA analysis are generally retained in our PDX xenolines that are serially passage in mice. So our tumors tend to resemble, to a very great extent, what is seen in patient tumors. And so it's a way of maintaining the cell line or the xenoline without having to go back on a regular basis to the patient and get the same tumor. We do see that the PDX have a few more copy number variations than are seen in the TCGA tumors. And so we're currently investigating as to what those really are and what they might mean and whether or not they have any importance in the assay. But more than likely, it's related to the passage in the mice and perhaps the selective growth that occurs there. So this is what the panel looks like now. We've got 27 that have been fairly well characterized. This is just a very, very small list of genes and other attributes that we've defined for these, but essentially we have neural tumors, we have proneural tumors, we have classical tumors, and we have mesenchymal tumors. And our tumors seem to look very similar to those you would find in patients. After all, they were derived from human patients. Now, the genomic profiling was quite a bit different in terms of what we wanted to do. The human kinome really sort of defines a lot of the enzymes that we call kinases that take phosphorus from ATP and they stick it on to a particular receptor molecule on a protein that either activates it or inactivates it. And we know that there are 518 known kinases of the protein kinase variety. 144 of those will phosphorylate a tyrosine residue on a protein to produce a phosphotyrosine, and 374 of those like to find motifs that have serine or threonine residues within those proteins. So there are a lot of proteins that get phosphorylated. There are over 20,000 sites. So these 518 kinases are pretty busy, trying to keep all of the proteins phosphorylated in the right order in the right time. One of the things this will allow us to do is to evaluate therapeutics in terms of their ability to inhibit kinases. This is a really big industry push right now. Almost every major pharmaceutical company is developing an entire panel of kinase inhibitors, these small molecule inhibitors that block, either very well or not very well at all, depending, block the activity of these kinases and push these through phase 1 and phase 2 trials. But they're basically throwing mud against the wall trying to figure out if it's going to work or not. They are not actually looking at specific instances where the kinase inhibitor might actually work. So by measuring kinase activity, we should be able to guide the selection of which kinases might actually be useful. So unfortunately, most of the clinical studies don't even bother to do that. They just see if they get a response or if the material is toxic. So this is the machine we use. It's a fairly inexpensive machine. It's only about $180,000. It lets you measure all of the kinases, the activity of all of the kinases in a very small amount of protein. It takes about 10 micrograms of protein, which is not very much at all. You can measure the kinase activity by applying the solution or the lysate to one of these dots. There are 12 of these that comprise the chips. Each one of the dots has a 20 by 20 array that looks at 400 different activities at one time. Each one of the dots is comprised of this collection of pores, and inside of the pores is the substrate that the kinase works on. And essentially by flushing the lysate back and forth up through the machine, and you've got an antibody in there that has a fluorescent tag on it, when you bring the material down towards the bottom and form a drop down here, you can take a picture of what the chip looks like, and you can measure in real time the ability of a kinase to phosphorylate a protein. And essentially by doing that over a period of an hour, hour and a half, you can actually get a kinetic curve that actually describes the phosphorylation, and you can convert that back into an equation that tells you how fast the enzyme is working, not whether it's working or not, but how fast it's actually working. So you can get real-time kinetic readout in terms of each individual enzyme. So if you take the data that you generate and convert it into a histogram, and here green is elevated activity, and red is lowered activity, and you look at our PDXs down here, these are the substrates that tend to stick out as being different. And if you just allow an unsupervised cluster analysis to be done, you can see that this divides pretty much into 3 separate clusters or 3 separate families and subfamilies of kinases that define each one of these PDX. So each PDX has its own signature, and some are like each other more than they're like the others in the entire panel. So by building this panel of baseline activity, we can determine how the kinases actually might work. So we can now add to our table a kinome profile. And we divided it into 3 different profiles and essentially call them 1, 2, and 3 for lack of a better term. And you can see that the classical is generally a kinome profile 1, but there are classical tumors that are actually profile 3. Mesenchymal cells are almost all 3. We do have one that's a 2. And so what this does is it adds a level of complexity and actually subdivides the different molecular subgroups that we have into kinase subgroups. The neurals are almost always 1. We do have a 2 down here, and the proneurals are always 2. So we have profiles that tend to be either classical or non-classical in terms of their interactivity. And by developing this particular profile, we have a way of measuring what's actually going on. So to give you an example, and one example, we looked at a drug called AZD1480. This is a drug that was produced by AstraZeneca. It was used to treat myeloproliferative disorders in patients. And it inhibits very nicely a kinase called JAK2. And JAK2 is very important because it phosphorylates a protein called STAT3, and when that protein is phosphorylated, it goes into the nucleus and causes the transcription of a lot of genes that actually are pro-tumorigenic. So by inhibiting JAK2, we thought that we could inhibit the growth of the tumor. And essentially what was done is that 2 of the tumors here are shown here. We actually treated 3. These are tumors that are put into the brains of mice, and then we treat the mice after a week with 2 weeks of oral therapy with AZD1480 or with the vehicle in which 1480 is actually dissolved. So we have 2 groups of mice, and then we follow their survival. And what you can see is that for this particular tumor, it appeared to be pretty sensitive to the effects of the drug. We had mice that were treated with the vehicle that died in a fairly reasonable order, but we had mice that even after, for a couple of 3 weeks after the treatment was stopped, they were still alive, and then they started dying, but only 50% of the mice died. So the other 50% were cured. We waited 6 months and finally got tired of feeding these little guys, so we killed them, cut their heads off, and looked in their brains, and we could not find any tumors. So essentially these were cures. This particular tumor, done the same way, put in the brain and then treated, you can see that it has some sensitivity, but we were still treating when mice were dying. So clearly this was not as a sensitive tumor. This was a less sensitive tumor. I wouldn't call it resistant, but I wouldn't call it so less sensitive seems to be a reasonable term. And by looking at a 3rd tumor, we found that this 3rd tumor behaved very similar to this one. Not quite as well, but very similar, more similar than this one. And so the idea then was, okay, we've got this great database of kinomic activity. Why don't we go back and look at our database and see if we can associate any of the kinomic activity with the response that we see in the mice. And so the first thing to do is to remove all of the kinase activities that are very similar for all 3 tumors. So we get rid of those, and we only look at those that are very different for the 3 tumors. This would simply be noise that would confound the cluster analysis. So we look at those that have a 30% difference from each other. And what we find is that the tumor that is less sensitive has a very different pattern than the two tumors that were more sensitive. So they cluster together, and this guy is separate. So this is a heat map that just shows you the activity. So 1016 was the less sensitive tumor. That's the one that you saw on the right-hand survival curve. And 1046 was on the left-hand survival curve, and 1066 I didn't show you. So then the question we ask, well, that's interesting. What if we take these same substrates that we identified over here and applied it to the rest of the panel? How would they cluster out based on those substrates? And what you see is that when you do that, you see that the resistant tumor kind of clusters with these guys down here, and the two sensitive tumors cluster with these guys down here. So on the right-hand side. So essentially what we're able to demonstrate is that even though we didn't test the other tumors with the enzyme, they would likely respond to it in the same way that the tumors that were actually tested. So to go back and do this for all 24 remaining xenolines would cost about $250,000. And I didn't have $250,000 to actually test these to show that this was actually accurate, but what I did have was the ability to take the same drug and the protein that we had used to generate all of this data, mix those together in the PAM gene system, and see how well the enzyme inhibited the same types of kinases. And so that's what we did. We did what is known as an ex vivo profiling, in which we're actually profiling a tumor, and this could be a tumor from a patient, for example, profiling that tumor by adding the kinase inhibitor. There's no kinase inhibitor here, there's one here, and you can see the drug knocks out a lot of the signal that's seen for many of the enzymes that are, many of the substrates that are defined on that single chip in that 20 by 20 array. And so that is essentially what we found, is that if you look at the tumors again, and we just looked at a small number of these because each one of these arrays is about $1,000. So I had enough cash to do 20 of these. So essentially the less sensitive tumors still associates in terms of its response to the drug in the ex vivo assay as the tumors that it associated with in the previous analysis we did. And again, the more sensitive tumors associated with the tumors that they associated with originally in the other assay, suggesting that if we indeed use something like this, we could take a patient's tumor, compare it in terms of the baseline, compare it in terms of its response to a particular kinase inhibitor, match it up with one of our tumors and see whether or not it would be resistant or sensitive to this particular therapy, and make a treatment decision within about 4 hours. It takes about 4 hours to make one of these runs. So you could get a piece of fresh tissue from the OR, and before the patient wakes up in recovery, you've got an answer as to whether or not the patient would be eligible for a particular therapy. That's expediting the decision-making process in a very real fashion. So our animal model system, our avatars, look like human GBM tumors. They have a lot of the same characteristic features that you would expect them to have and hope that they would have. We can use data analysis to show how they're more like or more different from human patient tumors. We can use genomic profiling as a very rapid means of looking at small molecule inhibitors that are supposed to target kinases. It doesn't do any good for drugs that work by other mechanisms. And then genetic profiling of patients' tumors, what might be able to be used for testing other things. And one of the other things that we're looking at are viruses. We're very much interested as why some patients respond to viruses and others don't. And just to show you where we are with this particular aspect, we're looking at Viruses that can be used either as a direct oncolytic agent, in other words, they infect a tumor cell specifically, they don't infect normal cells, they kill these cells, or we can put into the virus a gene or a series of genes that actually makes them much more effective in terms of, say, inducing an immune response by putting in a cytokine that activates the immune system in a very specific way. So the types of things that we've done with viruses are really based on work that was done many years ago, first working with non-replicating viruses, and then eventually, once we determined that we're not going to kill anybody with these by putting viruses into their brains or into other parts of their body, we went to replicating competent viruses that had been changed in some way to make them safe. And the virus that we're working on is Herpes Simplex. It's the virus that causes a fever blister. If you get herpes encephalitis, you're going to be dead in about 4 or 5 days. So it's a very dangerous virus to stick into the brain. On the other hand, we've been able to engineer Herpes Simplex virus by removing 2 copies. It has 2 copies of a single gene that we call the neurovirulence gene. It's the gene that allows the virus to grow in brain cells with reckless abandon. And this gene is called, I guess, gamma-1 34.5, basically because it's produced late in the virus cycle, and the molecular weight of the protein is about 34.5 kilodalton. So that's how it derived its name. In Europe, they call it RL. But the genome of a virus, of a herpes virus, is really pretty simple. It's got a unique long segment that has about 40 genes or 45 genes in it. It has a unique short segment that's got about 30 genes in it. And then there are a series of genes that are found in these segments, which are very important to the virus, so important that they actually put in 2 copies of each one of these genes in what are known as the terminal repeats and the inverted repeat. And essentially, those genes are really the driver genes, and that's where 34.5 resides. It resides in one of the terminal repeat areas and in the inverted repeat area. So we can actually make this virus safe by removing 1 gene, both copies of it. And a way that this works is that if you have wild-type herpes virus infection and replication, essentially what happens is that the host response attempts to protect the cell. And the way it does that, it has a mechanism for detecting double-stranded RNA and double-stranded DNA, and it activates this PKR response, which actually phosphorylates this particular protein. And by phosphorylating E1F2 alpha, it can inhibit the translation of proteins. So the cell can't make proteins, but the virus can't make proteins either. So the idea being that the cell will just sit there and wait out the virus. But what happens with herpes is that you have a situation where gamma 34.5, if it's present, will actually associate with a phosphatase, and this phosphatase will come in and dephosphorylate that protein and restore protein synthesis so virus can be made. So that's how the virus gets around the host mechanism. By taking out the gene and producing a 34.5 deleted variant, then we've removed this protective mechanism or the ability of the virus, rather, to infect normal cells. So normal cells are perfectly protected. They can use this intact system for shutting down the virus. So even though the virus gets in, it's not going to shut down the cell. Tumor cells have outsmarted themselves. They've defeated, they produce an activated RAS or a mutant RAS in the tumor cell to shut off this response, because this response could actually inhibit the growth of the tumor cell. So it wants to shut this off, so it produces RAS to shut this off. Well, that really plays into our hands very nicely, because then all tumors are sensitive to this particular virus, which can actually infect and kill them because this is shut off. But in normal cells, RAS is not activated, and so the protective mechanism is there. So we've built a virus. We've done a number of different studies with the virus to show that it's safe, We have actually done 3 phase 1 clinical trials with this virus in patients. We haven't killed anybody. The first trial was just to inject the virus into the tumor stereotactically and see what comes out. Essentially, we had 8 out of 20 responses to this particular single injection of the viruses. We had a few severe adverse events, but again, and these are patients who have a brain tumor, and the adverse events were related principally to their tumor and not to the virus. We decided to take a look at how one can actually measure what's going on. So we designed a second trial that actually had 6 patients in it, and it was designed principally to obtain tissue from the patient and to determine what's going on. Essentially, we had heavily pretreated patients that had no other option, but they needed a surgical resection to reduce tumor burden. So these patients were all candidates for resection, that the resection itself would provide a benefit, and by putting in the virus and then doing the resection and looking at the tumor that was resected, we could make some decisions about what was going on. So this is how it was designed. The patient was injected intracranially right through the skull into the tumor bed, and then 2 to 5 days later, the neurosurgeon came back and resected the entire tumor, again with the catheter in place so that we knew where the virus was injected. This is an example of a tumor with a catheter. You can see the catheter going through. It was pushed through so you could see the tip right here. We simply sliced this tumor and then quartered each one of the slices and did a variety of different studies. And the thing that popped up the most was that we had a very florid immune response to this tumor. Now, this tumor doesn't carry, this virus doesn't carry any particular immune stimulant in it, any cytokine, but the patient responded quite nicely to the virus itself and produced a very florid CD3 response, and then even more important to us, the CD8 population of T cells is the killer population, and we had a very nice response, again a focal response in the area where the tumor was injected. So this suggests that and these patients did much better than you would anticipate in terms of their survival characteristics after this single therapy. We've analyzed the tumor and we've identified about 49 genes that are correlated with survival. So these are all up in the patients who survived out to 20 months, and then the same genes are all down in patients who survived only 3 or 4 months. So we had a very clear description of how the gene expression panel might actually identify up front which patients would be eligible for this therapy, those likely to respond, and which would not, those likely not to respond. So we've have produced a virus that is fairly safe to use. We've demonstrated that we can get pretty good survival. Virus infection seems to work quite well. The question is how to make it more effective. We could use adjunctive therapy like radiation. We could augment the anti-tumor effect with immunotherapy, and we could develop a gene profile to actually screen patients and determine who's working. And so we've got a new virus that we've made. It's an IL-12 expressing virus. IL-12 stimulates the T cell immune response. We can see vastly improved differences in terms of survival of the mice and tumor growth. These guys survived out. These are all mice with no tumor after being treated with our new IL-12 virus. And essentially, we see the same type of response with the virus in the sense that we see a very nice florid response with both CD4 and CD8 killer T cells in the tumors that appeared within the first and analyzed within the first week or 2 weeks or so of receiving the virus. So we think that this particular virus will be even more effective. And we've completed our toxicology studies. We used 36 Aotus monkeys. These are New World owl monkeys, I suppose appropriate for Alabama. They're called redneck owl monkeys. But essentially these monkeys were not affected by the virus, so we think it's pretty safe. We got the RAID program to actually produce the virus, a fairly large number of vials. We have over 500 doses of this particular virus. We have both IRB and FDA approval for this particular virus. The virus is now sitting in the UAB research pharmacy, and we're waiting for the company that makes the unique catheters we will use to get their catheters approved by the FDA, and then we can start our trial. So that's essentially where we stand with our ability to select patients based on our PDX models and the studies that we've done in the patients with oncolytic virus. I'll be glad to accept any questions. Again, these are the people who did most of the work.
Thank you, Dr. Gillespie. I think we're due for a break now for 15 minutes, and if you have questions for Dr. Gillespie, certainly available during the break, but we'll also have the panel discussion. Sorry? Oh yeah, make sure you start collecting your questions so we'll have a good discussion coming up here. And again, thank you, Dr. Gillespie, for this, for that talk. So please, let's see, it looks like about 3:25 according to the clock in the back, so let's try to be back in 15 minutes, and we're okay for time. Thank you all.